Okay, 3, 2, 1, and we're live.
Okay, good morning, everyone. I think based on the timing, 12 on time, but we may want to, wait for maybe 1 or 2 more minutes just to make sure that everybody is in the room, and then we start our exciting session. Just want to make sure that, everybody is in the correct room. So please, pay attention to the title. You are in the symposium about, okay, so let me go to the correct opening slide, and understanding mesoscale visual processing through the lenses of high resolution functional MRI. There is another session, ongoing. So just make sure that you are in the correct session, please. We still have 1 more minute, and then I will start. Okay, so it's 12:02 and let's start. So good morning, good afternoon, good evening, good night, depending on where you are. Thanks for attending this symposium. My name is Shahin Nasr, and it's my pleasure to be your host and to open this symposium. So we usually start our session with a joke or a sort of, icebreaker. Believe me, I tried to find one. But everywhere I looked, I just found sad news. And I think you all know about shooting in schools, wars, natural and unnatural disasters. That's all that we found these days on news. And so I thought to myself, let's start one single VSS session without a joke. It's not going to kill us. And at the end of the day, we are all here to learn about vision. And by definition, vision is the ability to use your eyes and to see and interpret the world around you. The world around you. And unfortunately, today, the world around us is a very, very sad place. Hopefully one day, soon, we will learn to be genuinely happy again, but till that day. Okay. So after this, somber opening, so let's talk about brain again. And as you know, we can study brain in very different levels, including macro scale and mesoscale, levels. And for many, many years, people have spent time, to study it, in macro level. But what is the macro level? So in macro level, we usually, segment our brain based on, different criteria. And for example, here in 1909, Brodmann used, a psychoarchitecture to, segment brain into different sections up to 52 regions. And the segments, the point is when it comes to macro scale, segments are relatively large. And other people have used other criteria, like, for example, neuroanatomic landmarks or functional connectivity, or other criteria. And in all of these cases, we see that, there are relatively large segments there. In this level, usually inter-individual variability is small and ignorable. And thanks to that, we can do group averaging, we can use probabilistic labels and things like that. But more importantly, most of the regions of interest that we are dealing with in this level, we consider them as homogeneous, units. And because of that, we can do a smoothing and we can average the activity within them. But when it comes to mesoscale neuronal processing, for example, here in two individual monkeys, when we look at the first visual area V1, and we look at ocular dominance columns, you see that, there are some level of, similarity between these distribution of these ocular dominance columns. But at the same time, there is a very obvious difference between the two cortices. You see blind spot in both individuals, monocular crescent in both individuals. But when you look at, the pattern of ocular dominance stripes, it's a very different between the two. And based on that, you have the chance to highlight, the difference between individuals. But to do that, we typically rely on high resolution, high spatial resolution based on using relatively small voxel sizes. To do that, we usually rely on ultra high field scanners. Unfortunately, to this day, there are less than 100 scanners available to us around the globe. And compared to the number of 3D scanners that are out there, this number is not really high. But the question is, why do we use ultra high field scanners? The reason goes back to the size of voxels that is necessary for our type of studies, mesoscale studies. When we take the resolution, high enough based on using small voxels, for example, to this level, then the level of signal to noise ratio in image level or the temporal signal to noise ratio goes drastically down. And to compensate for that, we prefer to use higher or stronger scanners compared to scanners that work with lower magnetic fields. As you can see here, there is a significant amount of enhancement based on using 7T compared to 3T or say 1.5T, both for image SNR and TSNR. And but at the same time, just don't forget that you can compensate it in other ways, like for example, collecting more data. And when it comes to that, you have to consider that you need a huge amount of budget. For example, if you want to do that based on 3T and you want to collect a color selectivity map, in 7T with one session spending almost this much money, you can generate a very strong color selectivity map in area V2 and V3. But to collect something comparably to this in a 3T scanner, you have to scan your subjects 4 times. And you can see that gradually by averaging over these sessions, your the color selective activity appears. But even after 4 sessions, for multiple reasons, your map is still weaker than what you see in 7T in just one session. So there are big advantages for having a 7T. So to some extent, you can do it with 3T, but it always comes with a very, very large bill. One question that you may ask at this point is that when it comes to safety, is 7T safe? Because magnetic field is much stronger in 7T compared to 3T. Since 2017, First, 70 scanner, designed by Siemens company, the Terra series, they received their FDA approval. So it means that 70 magnetic field by itself is not dangerous to humans. So in that sense, if you have a 70 scanner, please be very careful about, safety, concern. But if those safety concerns are addressed, then there is no harm in putting your subject in that magnetic field. All of these said, so now back to our symposium. This symposium consists of 4 different talks. First, I will talk about visualizing cortical columns within the retinotopic visual areas V1 to V3 in humans with normal and amblyopic vision. And then Julia will talk about layer-specific profiles of factual and counterfactual feedback signals in human early visual cortex. After that, Luca will talk about characterizing top-down related functional microcircuitry of face processing in visual cortex. And at the end, Roger will talk about columnar encoding of personal space and visual space in human parietal cortex. Each talk will be something close to 20 minutes. We will have 5 minutes of Q&A at the end of that. And if you have more questions beyond that 5 minutes, then you can ask the questions in that public gathering room, and just make sure that you are going to area for Zoom room 2. Sorry. And we will be there and you can ask your questions. So no problem with that. So all of these things said, so now we will start our first talk. And it's about visualizing cortical columns in individuals with normal and amblyopic vision. Before starting my talk, I want to thank Bryan Kennedy. And my friend, without you, this wasn't, doable. We scanned something like, 500, sessions during the COVID time. And if this gentleman wasn't helping me, it was impossible. So thank you, sir. And he is applying for PhD positions this year. If his application comes to your desk and you reject it, there is something significantly wrong with you. Okay, so as an introduction, so I think, for more than 50 years, thanks to studies by Moncassel and also Hubel and Wiesel, we are aware of cortical columns. They are considered elementary units of organization in the cortex, and they are extending through all cellular layers in the cortex. And these units contain neurons whose salient physiological property, for example, stimulus selectivity, are more or less similar to each other. And unfortunately, to this day, evidence for cortical columns is mainly based on animal studies. And direct evidences in humans, they are relatively rare. And this is mainly due to a small size of columns in human close to 1 millimeter, compared to the size of voxels that we use in conventional fMRI studies, which is either 2 to 3 millimeter. And on top of that, we usually use, a smoothing that makes the data even, makes accessing cortical columns even harder. But hopefully during the course of this talk and also by other speakers, we will convince you that this is, this can change. And in this talk, I will provide you evidences of, 4 different types of cortical columns in humans in vitro. They include ocular dominance columns in V1, motion and stereoselective columns in V2 and V3 thickest stripes, and color selective columns in V2 and V3 thinnest stripes. And then I will show you how amblyopia is affecting their function. And during the course of my talk, I will present you data that is based on scanning using a 70 Siemens scanner, and they're all based on using a voxel size equal to 1 millimeter isotropic. TR was 3, TE was 28, and I used partial Fourier of 7/8. And all tests were done based on a block design. And subject task during these tests was color change detection for the fixation spot or something similar to that, completely orthogonal to the stimuli that we had on a screen. And during the course of pre-processing and post processing, we limited our analysis to deep cortical layer, the deepest 30% of cortex, and that was to avoid pial veins and artifacts attached to them. And we used the radial smoothing unless otherwise is mentioned, just to enhance our contrast to noise ratio. And okay, so let's jump on, ODCs. The first type of columns that I want to talk about here, you can see evidence for ODCs in monkeys and in humans based on histology and using cytochrome oxidase. And as you can see here, in both species, you can easily detect the blind spot, monocular crescent, and you can see stripes that are responding to contralateral and ipsilateral eye. And as you can see, detection of these stripes is typically easier around the foveal portion of visual field, and they are more differentiable from each other in both species. But when you go to the peripheral part, the area is mostly devoted to contralateral eye, and especially in monkeys, as you can see here, there are more like separate islands devoted to ipsilateral eye. But what about when the subject is alive and in vivo studies? So here you can see one of our studies. We published a preliminary result of that in 2016. Here you can see two different individuals. The activity maps is based on a stimulation of right eye versus left eye, both of them around V1 region. When we zoom in and we show the borders really, generated based on separate scan sessions, retinotopy mapping, you can see that the pattern of activity is mostly limited to area V1 in both individuals, and you can see a sort of a stripy pattern here. To facilitate this presentation, we usually cut our slices at the center of V1, and then we flatten them, and then you can see them like this. Right eye versus left eye stimulation. One of the subjects was right eye dominant, the other subject was left eye dominant. So when we do dominant eye versus non-dominant eye, you just see that the color is reversed in one subject but not in the other one. But the point here is that the pattern of stripy activity that you see here is mostly limited to V1. We see some sporadic activity outside V1, but the pattern is completely different than what we see in V1. And we also know that this pattern is expected to change in amblyopia. But you may say that, okay, so what is amblyopia that you keep talking about? For those of you who are not familiar with amblyopia or lazy eye is a developmental disorder caused by unbalanced binocular visual input early in life. It usually happens in the first few years of life. However, it's not rare to see amblyopia in later stage of that, but comparatively, it's more common in earlier stage in children. There are two common causes for amblyopia. One is strabismus, when one eye is deviated relative to its normal position, and the other one is anisometropia, when one eye shows lower refractiveness compared to the other eye. Amblyopia is associated with a long list of visual impairments, including decreased stereo acuity and impairments in encoding of high spatial frequency components. When it comes to neuronal level, studies in monkeys, for example, this one by Chiopis et al. In 1998, they have shown that amblyopia affects distribution of ocular dominance columns. For example, when you look at a monkey with normal vision, you can see a sort of uniform distribution of neurons that responds to contralateral versus ipsilateral eye. But when it comes to a strabismic monkey, you can see that this skewness has changed to shift toward the end of the spectrum, toward the amblyopic eye and non-amblyopic eye. That when it comes to anisometropic monkey with anisometropic vision, this shifts significantly toward the non-amblyopic eye. Can we see something like that in amblyopic humans in vivo? The answer is yes. This is the data from a control subject, location of fovea, and distribution of ocular dominance columns, as I showed you in the previous slide. This is one individual with a strabismus. As you can see, large patches similar to what you saw in monkey, that they respond to end spectrum amblyopic and non-amblyopic eye. When it comes to anisometropia, the activity is mostly dominated by the stimulation of non-amblyopic eye rather than the amblyopic eye. And there is one very, very obvious, other difference relative to control, and that's extension of ocular dominance activity toward area V2, V3, V4, V3A that you don't see typically in controls. So the first point dash, we can detect ODCs in normal condition and also in strabismic and anisometropic visual conditions. But what about, what beyond, the V1? We are also aware that, in area V2 and V3, there is similar type of, heterogeneity. In V2, in both humans and monkey, studies based on cytochrome oxidase staining has shown that there are different stripes, that they differ from each other in their shape and the level of myelination. Based on that, and based on mainly in the data that comes from monkey, we call them thin and thick stripe, and you also see the pale stripes between them. In monkey, originally it was suggested that it's mainly in V2, and they couldn't, differentiate, or say anything about V3 because monkey V3 is very, very small compared to what we see in human. But at least in human, we see a very clear extension of stripes into, area V3. And one other difference that you see between monkey and human is that, it's really hard to say which one is thick, which one is thin. And the shape is not really a factor here. When it comes to their selectivity, we already know that when we compare the stimulus selectivity, in this case, color selectivity, in this case, orientation selectivity, relative to location of, these stripes localized based on cytochrome oxidase, you see that thin stripes, they show color selectivity, and thick stripes and pale stripes to some extent, they show orientation selectivity. Other studies, for example, studies by Anna Rowe and others, they have also shown motion and stereoselectivity within thick but not thin stripes. Can we do the same thing in human? This is the data that we see. When we look at, the functional localizer based on colorful versus luminance varying stimuli, we see that strong activity in V1 as expected from studies, single cell studies in animals. This is data from one single human subject. You can see that in V2, you see stripy activity perpendicular to V1, V2 border. In the whole V2, extend to V3, we see some activity in V4, and we see area V8. We don't see that much activity in area V3A. When it comes to stereoselectivity, we see a very beautiful map of stereoselectivity in V2, some activity in V1, again, consistent with data based on physiology, electrophysiology, extension of activity to V3, very strong activity in V3A, V4, some activity around area MT. And when it comes to motion selectivity, you see this activity again in V2, extension to V3, strong activity in V3A, and extends to MT. Can we localize these stripes based on other type of stimuli? It appears that this localization is independent of a stimulus shape or 2D structure. For example, you can localize motion selective stripes based on concentric rings or moving random dots. Pattern of activity remains the same. Again, this is the map based on one single subject. And here you can see a stereoselective activity based on random dot stereograms or disparity varying grading. Again, you can see that the pattern of activity remains the same. So localization of these stripes appear to be independent of a stimulus 2D structure. And are they overlapping or non-overlapping? Data suggests that when it comes to the level of overlap between these stripes, they are mostly non-overlapping, despite their small size and the fact that they were collected during different scan sessions. When we put these maps on top of each other, we see that they're mostly non-overlapping. This is the data based on one subject that I showed you before that. I showed the map, put the map on top of each other. You can see that in V2, V3, and V4, the stripes are non-overlapping. The overlapping parts are presented in gray. So you see that the size of overlapping part is small compared to non-overlapping section. And here you can see the same data in 15 other hemispheres. So this is something that you can repeat across different individuals. One question is, because especially in monkey studies, people prefer to also check orientation selectivity. The pattern of orientation selectivity in humans is relatively weak. So I did a ROI analysis over data from 15 different individuals. And as you can see, consistent with the data in the monkeys, stereo and motion selective stripes that are believed to be parts of thick stripes, they show higher orientation selective activity compared to color selective stripes that are believed to be a part of thin stripes. This is exactly what you see in monkey studies. And you see the same pattern also in area V3. If you're interested in this topic, please read our preprint. It's already under review. Hopefully, fingers crossed, it will be published soon. But one legitimate question here is that, okay, dude, you're talking a lot about, all of the, these patterns of activity, but are you sure they are columnar? So there are multiple ways to convince you guys to say that, this is columnar. One way is to show you this activity across different layers. For example, here, John Polimeni et al., they did something like that. They showed a sort of, M-shaped, on a screen, and then they sampled from different layers of V1. As you can see, in deep up to superficial layers, you see the same pattern. The only difference is that when you go to superficial layers, the pattern becomes slightly blurred. That is believed to be because of impact of Piolle veins. But when it comes to our data, again, when it comes to, for example, stereoselectivity, you can see the pattern across deep, middle, and superficial layers. But you can also quantify it by showing that if you measure the correlation within a column between superficial relative to deep layer compared to across columns, within superficial or within deep layer, but outside column, then this correlation should be stronger within column rather than across column. This is what you see in V2 and V3. The level of correlation is stronger within a column between superficial and deep rather than between deep and deep when they are across column. The level of correlation is higher here. But what happens in a case of amblyopia? In case of amblyopia, we expect the number of stereoselective columns to go down, while color and motion selective ones are expected to stay high. Here you can see color selective activity, it's still there. Motion stereoselectivity, this subject is stereo blind, consistent with that. there isn't that much activity here. So it's not just disparity, it's also the sense of stereopsis. And when it comes to motion selective, again, activity is there. Is interdigitation still there? Yes, interdigitation for motion and color that are still, detectable. It's still there, and you can see that here. Again, if you are interested in this topic, please watch our video from last year. We assessed that we showed this data in more details. But what will happen to the size of the stripes? As a matter of amblyopia, we believe that the size of stereops and, motion and color selective stripes go up compared to what we have in controls. So technically, rather than showing no selective activity, we are seeing, larger motion and color selective stripes, while the number of non-selective, locations remain the same. And that supports the idea of plasticity during the course of amblyopia rather than atrophy. And how about, spatial frequency or shape encoding? Here we provide data that when it comes to stereo and motion that are parts of magnocellular stream, they prefer, lower spatial frequency components. As you can see here, a stripy pattern of activity in response to lower spatial frequency component. And when it comes to color selective ones, they show more response or more preference toward higher components. If you want to know about impacts of amblyopia on that, please watch our talk in ARVO 2022 and also in VSS, it was in person, but I think the video is now available, or follow us on Visual Vision, YouTube channel. I think we will upload them hopefully by the end of this week or maybe next week. And as a summary and conclusion, I just say, hopefully I have convinced you that we can now localize at least 4 different types of, cortical columns in human V1 to V3. And hopefully I have convinced you that we can detect impacts of amblyopia on this functional organization. And again, hopefully I have convinced you that fMRI these days is a reliable tool for visualizing, localizing, and also studying the columnar organization in human visual cortex in vivo. At the end of my talk, I want to thank, Peter Becks, Eric Geyer, David Hunter, Roger Tutel, my dear collaborators, and also our funding agency was very generous of them, NIH, Mass General Hospital, and Boston Children's Hospital. And last slide, please visit our website. Please contact us if you need any data from our studies, any code, any advice on data analysis. And for younger audience, if you're looking for a postdoc position, please contact us and send me your CV, and we'll be more than happy to chat with you about that. Thanks a lot. I'm ready to answer your questions, and I will stop sharing to be able to see your questions.